Okay, so going through the multiple choice questions for the cytology topic, the integral proteins of the plasma membrane interact with. Remember that the integral proteins are what make up the lipid bilayer. They are always found on the periphery, like the peripheral proteins, but these are more common than peripheral proteins. Here you can see an integral protein which looks a bit like transmembrane, ignore that, but the integral protein is usually like basically on the side like this. And what I've tried to show in here is a peripheral protein bounded to the integral protein. Therefore, the integral protein does interact with peripheral proteins and it interacts with components of the cytoskeleton. It cannot possibly interact with nucleolus. This is found within the nucleus, and these are the organelles. Glycocalyx, we've already talked about glycocalyx being basically a glycoprotein coat made up of carbs and protects the cell immune function. It does take part in cell adhesion, does take part in cell cooperation, and it is a polysaccharide layer. It does not take part in ATP. These are irrelevant for this. Next. So the types of adherent junctions, you've got desmosomes, hemidesmosomes, and zonular adherents. What is a nexus? So nexus is when there's two cell membranes and two structural units, and they are combined with a sixth structural unit made up of nexus, which is made up of connexomes. And this forms a gap junction. Whenever there's a gap between two things, it's allowing, for example, oxygen or substances to pass through this gap. That's what's happening here. There's passage. The gap is created, allows for substances to pass through. So it's built of connexomes. Yes, it is not a cell organelle. It is not an occluding junction. It's nexus, connexone, gap junction. It's a gap junction. Consisting of protein channels for transport of small molecules and ions. Yes. Enzyme, no. Basophilia of the cell cytoplasm. Basophilia is always to do with ribosomes. Just remember that. So it is the presence of abundant rough surface endoplasmic reticulum, because they are the ones that contain ribosomes, not SCR, numerous ribosomes. Nuclear pores. Now, nuclear pores are, here is the structure of a nuclear pore complex. The nuclear envelope is the barrier between the nucleus and the cytoplasm. And the nuclear pores are the gateways across that barrier. Replication and transcription of genetic material within the nucleus requires the participation of proteins, and nuclear pores allow for this to occur. These are localized. If they would be localized just on the inner nuclear membrane or just on the outer nuclear membrane, how exactly would it allow things to pass? So these are not true. Function is selective transport of substances through the nuclear envelope. Yes, it is selective. Are formed at sites where the inner and outer membranes of the nuclear envelope are joined? Yes. Are built connexones? No, because nuclear pores are not gap junctions. So it's just two correct here. The nucleolus. Now, nucleolus is this. Here's the nucleolus. The nucleolus is found within the nucleus, and the ribosomal subunits exit the nucleolus into the cell where they combine with other substances to become complete ribosomes. So, nucleolus is where ribosomes are stored and where they come out from. So, general cell organelle, no. We've already discussed how general cell organelles are already. They are question 57. They are mitochondria, endoplasmic reticulum, Golgi, lysosomes, peroxisomes, coated vesicles. So this cannot be a general membrane cell organelle. It is related to the formation of subunits, yes. Enzyme, okay, sorry. Yes, it's not a general cell membrane organelle. It is built of parts of the chromosomes 13, 14, 15, 21, 22. It is a place where the ribosomes are formed. It is a component of the nucleus, and it's not bounded by a membrane. The main function is smooth surface endoplasmic reticulum. Formation of secretory granules? No. Adds carbohydrates? No. It's the synthesis of lipids and steroid hormones, and the synthesis of glycogen and mucose and intracellular transport. Term dictosome. Okay, what is a dictosome? Here is a visual representation of a dictosome. Dictosomes are stacks of flat membrane-bound cavities or cisternae that together comprise the Golgi apparatus. So that's what makes up our Golgi. Here. Is it here? I think it's here. Okay. So, component of Golgi complex, yes. Component of centrioles, no, that's a different organelle. Flattened cisternae with outer forming, inner secreting, yes. And it releases secretory granules from the outer surface. Which of the following features are specific for mitochondria? Possess own genetic apparatus, formation of new mitochondria by budding or simple division to ATP synthesis. Those are the three. which of the following features are common for mitochondria and peroxisomes? Basically, mitochondria are one of the few organelles which do contain a double membrane. Here, you can see mitochondria, nucleus, and chloroplast. Mitochondria and nucleus are the only double membrane organelles here, of the obvious ones. Lysosomes, ER, and Golgi is single membrane. Okay, so then you know that mitochondria is, but peroxisomes isn't. Contain a matrix with numerous enzymes. Okay, mitochondria and peroxisomes, the one thing that they both have in common is that there is a matrix present. Here you can see that mitochondria has the inner and outer membrane, has a double membrane layer. There's the peroxisome. It contains a crystalline core, which is a crystalline matrix in the core, and this one also contains our matrix there. Okay. Are generals membrane cell organelles? Yes, we've already discussed them. Possess own genetic apparatus? No, only the mitochondria does. Which of the following features are specific for lysosomes? Intracellular digestion, hydrolytic enzymes related to cell aging and cell death, because when organelles are lysed or lysis takes place, they are replaced, and this is cell aging and death. Peroxisomes take part in stereosynthesis? No, that's SER. Take part in intracellular digestion? No, that's lysosomes. Contain oxidative enzymes? Peroxisome has oxidative enzymes. Contain matrix with crystalloid? We've seen. Phosphorating enzymes? No. Which of the following features are specific for microtubules? They sustain the cell shape. They are not just built from protein actin, they have other things. Intracellular transport of molecule organelles? Yes. Ensure mobility? No. Formation of spindle fibers? Yes. Finally, mitochondria is visualized using acid fuchsin and iron hematoxylin. Typical for nuclear membrane. It is double layered, because we've already seen that. It is continuous with the RER, and it does contain nuclear pores.